Il n'y a pas d'air conditionné. Ils vont dormir.
Si,
Si.
il y en a, mais je ne sais pas si bon.
Allô, allô? Allô, ¿está bien? Si? ¿Me oye?
Muy bien, oui. Ouais. Vas-y, continue.
Je continue à parler. Est-ce que vous m'entendez bien? Allô, allô? Allô, ¿está bien? Est-ce que vous m'entendez bien? ¿Me oye? C'est pas moi. C'est pas moi qui parle.
C'est l'écho.
Mais non, c'est même pas l'écho.
Ok. Ok. Tout ok.
Alors, je le
Tu veux une bouteille d'eau peut-être?
Hey, good afternoon. For this seminar, today we have a very special guest, and it is a great pleasure for me to introduce Professor Varenne. Varenne, she's a distinguished professor of chemistry at the National School of Advanced Chemistry of Paris, Chimie ParisTech, in France. She currently leads the CESAT laboratory within the Institute of Chemistry of Life and Health Sciences, ICLIS, and has been a pivotal figure in shaping chemical education and research at University Paris Sciences et Lettres and other institutions around the world. Professor Varenne has held numerous leadership positions, including Deputy Director and Dean of Education at Chimie ParisTech, and President of ECOS North for France Scientific Collaboration within Latin America. Her contributions to science policy and academic evaluation have stemmed through her involvement with French and international advisory committees, such as ICEERS, for evaluation of French laboratories and institutions, Euramet, the European Association of National Metrology Institutes, CONACYT, and the CSIC, the Spanish National Research Council, and others. Her research bridges physical, analytical, and bioanalytical chemistry with applications in microfluidics, sustainable materials, and biomedical sensing. She has supervised over 30 PhD students, including me, and 8 postdoctoral researchers, and published more than 100 research articles. Professor Varenne's contributions have been recognized with prestigious awards, including the L'Oréal Award, the French Analytical Chemistry Society Award, and the Palmes Académiques, which is one of the France's highest honors for academic service. So, in addition, I can say that she's an excellent and wonderful human being, and she's multilingual. So, if you have questions in another language, in languages other than English, for instance, French, Spanish, German, you can speak with her openly. So, please join me in welcoming Professor Varenne.
So, thank you very much, Gonzalo, for this very nice introduction. I hope you can hear me. Is it okay? Okay. So I will speak with you today about the way we deal with analytical methodologies so as to develop some tools, methods, so as to develop diagnostic or theranostic. I will explain you afterwards. So first of all, I come from Paris, and in Paris there are some universities. My university is named Paris Sciences et Lettres in French, Paris Sciences and Letters, which indicates that it is a multidisciplinary university, going from human sciences till engineering and art sciences. And for example, there is chemistry. There is a graduate school in chemistry, so I'm the director of this graduate school, and I'm very happy to deal with this graduate school that includes very old institutions, such as my institution, Chimie ParisTech, ESPCI, École Normale Supérieure, so very known and old institutions that make together the university. And so my institution, in French, École Nationale Supérieure de Chimie de Paris, which is in the center of Paris, in Quartier Latin, a very nice place to work. But I don't live there, I live in another place. So it is in the center. My group is synthesis, electrochemistry, imaging, and analytical sciences for diagnostics. So it seems quite complicated because we've got different expertises, but we share our expertises so as to design and develop diagnostics and theranostic tools through analytical, electrochemical, imaging methodologies. So we share our expertises so as to develop very interesting molecules and probes. So in synthesis, we develop classical synthesis, but as well microwave synthesis to generate nanoparticles. We develop continuous flow synthesis, so different methodologies. In electrochemistry, we develop electrochemical sensors and biosensors. And we've got a scanning electrochemical microscopy that helps to imaging by electrochemistry surfaces, as you can see here. So the other one is analytical systems. So we work, and I will discuss with you about this during this seminar, so I will not enter too much in details. We work also with microfluidic systems developments. And then finally, we've got an MRI apparatus in our lab, in our group. And so we can go to imaging with optical magnetic resonance imaging so as to go till the application for the theranostics or photodynamic therapy evaluation. So this is our expertise, and I will go more deeply in this one. So for analytical sciences, so we develop either in electrophoresis, I will explain you what is electrophoresis, or developed in microfluidic formats. And we work on different applications going to interactomics, proteomics. Sorry, it does not go very quickly. So we work with different things so as to develop diagnostics and theranostics. And so we work on this domain so as to, and today I will show you how electrophoresis, which is a separative methodology, helps to design tools for diagnostic therapeutics and theranostics, which is the combination of therapeutic and diagnostics. So theranostics is the main objective, and I will explain you what are the properties expected from nanoparticles so as to gain in theranostics. So if I want to show you a little bit, not my plan, but what I will explain to you, is how can I go from electrokinetic methodologies to develop diagnostic tools? So for developing such diagnostics tools, we have to find new chemical, biological tools that will help for targeting, drug delivery, and imaging. So for this one, we have to find selective ligands that can help to capture a molecule of interest, a biomarker or a receptor. And we have to find, in some cases, nanoarchitectures so as to make a delivery in the body till the organ of interest. So when we deal with these diagnostic tools and systems, we are first to design, which type of nanoparticle we want to obtain. We have to do its synthesis and functionalization. Then we go to physicochemical characterization of the in solution. And then we go to an understanding if the nanoparticle can deliver a drug and in a very targeting phenomenon. And for all these aspects, that I cannot show you because it does not work, but on all these aspects, I want to explain to you that electrophoresis is a very interesting tool so as to help to go in a very good investigation and finding the nice systems. So what is electrophoresis? Maybe you don't know. You may know about chromatography, which is a classical separation method. Electrophoresis is a totally different method, and I will explain you why it is more interesting for me than chromatography. So here you've got a big apparatus, which is this size. So it's classical capillary electrophoresis. And we work as well in miniaturized systems, microchip electrophoresis that we develop ourselves. We provide them, we construct them, and we design them as we want. So you can see it's like 4 or 5 centimeters long. So we work in both systems, and what is very interesting in these systems is that the channels that are either a capillary here or a small channel here, is that they are very small, from the 50 to 100 micrometer diameter. This is very interesting for us because it's more or less the size of a vessel. And if we want to mimic interaction in physiological conditions, we are in the right dimensions. So we can deal with this. Furthermore, the flows that we use in these systems are more or less the same of flow as for the blood. So we have systems that can mimic externally what we would have if we inject the nanoparticles in the body. So now going deeper to electrophoresis, I will not enter in the details. When presenting the results, I will not enter in the details, but you are free to ask me afterwards about this methodology. So what is capillary electrophoresis in a classical format? It's a capillary, which is 50 to 100 micrometer diameter, around 20 centimeter to 30 centimeter long, and we apply an electric field. There is no flow, no pressure, it's only an electric field. And the compounds, according to their charge and their size, will move at a given electrophoretic mobility. And this given electrophoretic mobility is peculiar of a compound in a specific liquid solution. So with this methodology, which is very simple to implement, which can be automatized, we've got different ways of doing this. I will not describe them to you. What we do classically is separation. We can separate compounds if they don't have the same mobility. And then we can quantify as well, if we've got the good detector that is in the good limit of detection. But what is more interesting for me is that it is totally free in solution. If you know about chromatography, in chromatography there is a stationary phase. Here there is no stationary phase, so everything is free in a solution. So I can, and I will show you, study the interaction between two compounds without interfering interaction. So, and there is no laminar flow leading to peak broadening, and then it does not allow to have a good, precise determination of the compounds. So thanks to these properties, we can do very fine physico-chemical characterization of nanoparticles. We are expert about this. We can study non-covalent interaction characterization. So it means an interaction between a compound A and B. So what does it occur with them? We can do kinetics, we can do thermodynamics about this, and so we can study more generally reactivity in solution. So for this, I will show you some examples that we do with nanoparticles, which is main the subject of this. So with nanoparticles, we can separate by electrophoresis nanoparticles that differ in 1 nanometer in size. So this is very precise, so a very nice separation in size. When we want to functionalize a nanoparticle with different moieties, such as amino moieties, then according to the rate, the concentration of amino moieties on the nanoparticle, we've got different electrophoretic mobility. So we can separate also in function of charge. And this allows us to help to optimize functionalization processes while choosing the number of amino functions on a nanoparticle. Then what we can do is study when I've got a nanoparticle that has integrated a drug so as to make a drug delivery. So we can quantify the drug loading and the drug delivery properties. I will show you an example if I've got time. And then finally, so as that these nanoparticles can be compatible, biocompatible, biodistributable in the body, so we have to study, for example, protein corona, which can impact the movement of the nanoparticles within the body. And so we can do it by electrophoresis. So you can see here main characterization that we can do. And if I can enter only a small details about the theory, when I want to study an interaction, I've got a substrate, a ligand, there can be an interaction with many ligands on a substrate. Everything is free in solution. I can calculate interaction strength and kinetics, the amount of substrate and ligand that make the complex through mobility shift and thanks to a good detection sensitivity. For example, here I can show you this example. Here you've got an increasing concentration of ligand on the substrate and you see a change in the mobility. From this change in mobility, it indicates that it has an interaction, S with L, and from this shift in mobility, we can assess to the binding constant. Here it's the same, but with a difference in concentration in this frontal analysis. Here you've got another example I will present you afterwards or even by incubation. You can see the free protein and the complex protein, for example. So all from the mobility of a compound or a complex, we can access to very precise interaction constants. So, for lack of time, I will not speak to you about which type of ligands and how we deal with ligands. We work mainly with aptamers, which are very interesting molecules. If you are interested, you can discuss about this. I will go directly to nanoplatforms. So in our lab, we develop different nanostructurations that we can integrate in diagnostic tools. We work with solid nanoparticles, like a magnetic iron oxide, persistent luminescent nanoparticle, quantum dots which are decorated with copolymer, with gold nanoparticle for cells, and we work as well with other structures which are not so solid, graphene quantum dots that are layers of graphenes. It can be random copolymer nanoaggregates, and it can be as well peptides. Peptides are very interesting in nanostructuration for therapeutic and diagnostic application because they are biocompatible, they are not toxic mainly. And so we can work on these systems and we can have peptide nanotubes. And I will show you an example about peptide nanostructurations to provide theranostic agents. So, when we want to develop theranostic nanoplatforms, which properties do we need? First, we have to characterize deeply the nature, so it is what we decide to do, the size of the nanostructure, and the surface charge, so that we can deal with the right properties that we want from the intrinsic properties of the nanoparticle. Then we can add, by functionalization, a specific ligand, so that it can help the nanoparticle to target the place in the body where you want to be active. Then you can add probes by functionalization that will be MRI or optic probes so as to do imaging directly in the body. And then we can find the structure so that it can be responsive to an external parameter. For example, if there's a change in pH, then the drug that is encapsulated in a nanoparticle can be released, and I will show you an example. So there are the intrinsic properties, and then we can functionalize so as to gain other properties that are very interesting for our objectives. And then when you go to the injection in the body, there are many parameters to study in terms of pharmacokinetics, so administration, biodistribution, metabolism, and clearance. And I will show you some example about protein corona, and I will go further in this. And for all this, I can use electrokinetic methodologies, and I will show you some examples. But one methodology is not enough. We have to deal with complementarity, with dynamic light scattering, transmission, emission, microscopy, zetametry, and I will show you the complementarity of these methods. So the main difference between electrokinetic methodologies and the other ones is that with electrokinetic methodology, I separate the mixture in my sample. When I do DLS, it's all the mixture that give an average indication. Here we can separate and then identify each of the nanoparticles that we did produce. So this is the main advantage in this system. Okay, so our objective in our system is summarized here. First, we have to make a precise control of the nanoplatform synthesis or formulation. And so for this, we can develop various processes of synthesis that can be hydrothermal, microwave, in batch, in milli, in microfluidic tractors. Then we have to control the functionalization so as to have targeting, imaging, and drug delivery, so all this. And then we go to fine characterization. With classical morphological and elemental analysis, as I said, TEM, zetametry, DLS, and our system with electrokinetic separation. And then when we do this characterization, then we can come back to the synthesis to improve the synthesis. So it is an iterative process. And then when we are ready with the right properties that we want, we can go to biological validation. That can be in vitro assays or in vivo assays, and I told you we've got MRI apparatus, for example, in our lab. All what we do in this part, and mainly from this, is to limit the use of animal testing so as to prove the usefulness of the nanoparticles. So for us, this step is very important. And so I will go to the first example. This first example is with persistent luminescence nanoparticles. They've got, I suppose many of you know about these types of nanoparticles. They've got a particular optical properties allowing their use for in vivo imaging. So they can be sighted in the tissue transparency window by visible photons, and then they can emit light for hours after the end of the excitation. So it's very useful when you want to make a diagnosis in a longer term than some minutes. To develop these nanoparticles, so we work with persistent luminescence nanoparticles made of zinc gadolinium chromium oxides, and this is the first step of the synthesis. As you will see in the next slides, this work was done by Gonzalo Ramirez. It was his PhD work that was done in Cotutela between France and Mexico. So I was very happy to have Gonzalo in my group. So first, the synthesis of the initial nanoparticles. They've got hydroxides on their surface. So as to gain in biocompatibility for these nanoparticles, one possibility is to do what we call pegylation. So to add a peg that makes a biodistribution of the nanoparticles in the body. So as to reach this, there is an intermediary step with a silanization with NH2, and then NH2 so as to gain the pegylity. So it's a two-step process so as to decorate correctly the nanoparticle so as to be biocompatible and biodistributable. At each step, we did different methodology, such as dynamic light scattering, laser Doppler electrophoresis, TEM, infrared, so you see some examples here, and as well with electrophoresis. What does electrophoresis give more information? For example, here you've got the initial nanoparticles with OH. OH in the medium of separation is negatively charged, so O-, and so we've got a mobility that is negative. When we do the first step of synthesis, we go to cationic nanoparticles, and we've got a positive electrophoretic mobility. And then we do the pegylation, pegyl is neutral, so we don't have any more mobility. And so from the electrophoresis, we can follow all these steps. What this information gives us is a very good monodispersity of the sample. And this is not really visible here and here. It's very visible here. We've got very monodispersed nanoparticles in size and in charge. Furthermore, this information provides us the hydrodynamic diameter and the zeta potential. So you see the complementarity between this methodology that does a separation, efficient separation, and that gives information that we can compare with DLS or laser Doppler electrophoresis. And we had the same results. So it proved that electrophoresis is very interesting for this. Then, as I told you, when we want to inject the compounds, there are different parameters to optimize. And concerning the biodistribution, the objective when we do with nanoparticles is to have the longer biodistribution in the body. And so biodistribution can be perturbed by protein corona. What is protein corona? It is when you inject a nanoparticle in the body, there are many proteins in the blood, and so they can create like a decoration of the nanoparticle. They can lead to nanoparticle aggregation, so it will not distribute. It can lead to other phenomena, and we have to take this into account. So we have to understand what is the protein corona with a nanoparticle. And for this, we do it with electrophoresis, so as not to inject in animal and test, but we do it externally. And so this is what we do. Instead of doing with in vitro and in vivo studies, we decided to go to ex vivo studies and to do it with electrophoresis. I will show you an example. For example, you remind the synthesis is in two steps. The first step is a silanization with amino function. And so we decided to do it with two different times, synthesis time, 3 hours and 5 hours. What is the difference then on the interaction in the body? And so these two types of compounds that were pegylated after this intermediate step were studied on animals, and we could see that they didn't provide the same biodistribution, and we didn't have, by electrolysis, the same binding constant. And so this indicates that the structuration around the luminescent nanoparticle is very important as well. So what is the impact of the core on the interaction with proteins? So this was made with BSA as a model protein. And then we went in very deep physicochemical characterization of the interaction by electrophoresis with a specific method that is called Hummel-Dreyer. I will not enter in the details, but only to tell you. I've got a capillary that is filled with a protein. Then I inject, for example, here first water, a short plug of water. What do I see? There is a depletion of proteins where the zone of water is. Then when I inject the nanoparticle interact with the protein, and then I see a deeper depletion of the protein, of the concentration of protein in one zone of the capillary. And this is seen here. Here I've got a flat line. Here I've got a depletion, and here I've got a bigger depletion with a positive peak that is the complex. So I can see the complex between the nanoparticle and the protein, and I can see the depletion of protein. That it means the quantity of proteins that was decorated on the nanoparticle. And from this value, we can do a different concentration of nanoparticles or proteins, and we can access to the binding constants and the number of binding sites. So we can do isotherms, and we can access to the binding constant and saturation degree on the number of nanoparticles on the number of proteins on the nanoparticle. And for example, we did it at different ionic strength. The higher ionic strength is one that is the nearest to the physiological conditions. And so we can determine the binding constant and the number of proteins per nanoparticle. And this is very interesting when we want to continue the optimization of the design. And from this in determination by electrophoresis, then we went to MRI so as to compare our results with what occurs in the reality in a small mouse, white mouse. And so what we did is compare the biodistribution of non-decorated nanoparticles with the biodistribution of the pegylated, that is decorated nanoparticles. When we want to develop these theranostic agents, we want a biodistribution that is the longer possible. And then when you see with hydroxylated nanoparticles, there is a concentration in the liver with a very short time. When we've got the pegylation, it allows the distribution of the nanoparticles within the body for a much longer time. As you can see, the blue is the distribution of the samples. Okay, so the pegylation increases their permanence in blood circulation. And this is very interesting so as to develop theranostic agents. In the second step, what we did is a toxicity study. And we could identify that with the hydroxylated nanoparticles, we had toxicity at high dose. And when we decorate them with PEG, we could have non-toxic. So we could compare the biodistribution from electrophoretic results, and they were totally coherent. And this is something that we cannot do by electrophoresis, so this is a complementarity that we can gain about toxicity. Then, this is about protein corona. In the literature, if you are aware of this, in the literature, it is one protein analysis at a time. So, but the blood is made of many, many proteins. And there is no efficient methodology that allows to analyze the protein corona with 2 or more proteins. And so with Gonzalo, we had the challenge to face this and to do it by electrophoresis. And so we chose 2 proteins. The first one is human serum albumin, which is highly abundant in plasma. And the second one is apolipoprotein A, which is associated with the active transport of nanoparticles through the blood-brain barrier. And through this interaction of the apolipoprotein, it can be a way to provide drugs within the brain, which is very complicated. And so we faced this challenge with HSA and APOE, and I will show you some different results that we had. Don't worry, I will not explain everything. But first, we studied the interaction with one protein at a time. With APOE, we've got a change in mobility. With HSA, we've got the depletion with a higher depletion according to the concentration of protein. So you see already only with two different proteins, we don't have the same behavior for the interaction with the nanoparticle. And this is very interesting. It can be related to different kinetics. So for HSA, the kinetic is quicker than for APOE. But then we went to mathematical transformation of the mobility and depletion, we can access to the binding constant. And what we did evidence is that the interaction with the APOE is much stronger than with HSA. HSA is very abundant in the blood. APOE is less abundant. But even though APOE has got a very high binding constant. Then what it is, the challenge, is make a coupling of the two proteins. And so we try to understand the protein corona leading with two proteins at a time. So as you see, it's very complicated. I will not show you and explain you everything, but what we evidence is first, the APOE is more stable even in the presence of BSA. So I mean, if I've got these two proteins, APOE is the one that will interact the most. That could be very interesting if we want to develop theranostics for the brain. The second one is that we evidence through these electrokinetic profiles that HSA and APOE interact together. So the proteins can interact together, and we evidence as well a ternary complex, that is, the nanoparticle can be decorated by both APOE and HSA. And this can be very interesting to go further in the biodistribution, biocompatibility, to gain more information about this protein corona. So, for this example, I did show you this example with information about physicochemical characterizations, so which is complementary to classical DLS, zeta, and TEM. So this is the first objective that we did have. The second one is to study the biodistribution and the protein corona. And then we did show that electrokinetic methodology can be very interesting to go further in the understanding of protein corona while reducing animal testing. This is, for ethical point of view, very important. So I did finish with this first example. Now we'll go, so you remember the nanoparticles are solid nanoparticles. I will show you now another example where we used, as we can say, soft matter. It means proteins, okay? So we work on proteins for, as I told you, to make nanotubes or to make spherical micelles. And proteins are already in the body, so they can be very biocompatible. And so we worked on this thing. Proteins can auto-associate, auto-aggregate, auto-assemble through weak interactions. These weak interaction, I suppose you can understand them. It can be hydrogen bonds, hydrophobic interaction, electrostatic interaction, PP interactions. And through the exact sequence of the peptide, one or many of these interactions can occur. Okay, so there are interesting factors, the number, the nature, and the sequence of the amino acids. There are extrinsic factors. It means this auto-assembling depends on the pH, temperature, nature, concentration of salts, nature of solvent. And so we have to deal with all this so as to make an auto-assemble. And this auto-assembly was already shown as nanotubes are spherical, nanofibers, micelles, etc. But there were still not studied for theranostic agents. I mean, if I got a peptide that auto-assembles and that can integrate a drug within this auto-assembly, then the drug can, the nanoparticles can deliver the drug in the body. And so we were crazy, so we decided we synthesize ourselves the peptides, okay? So we chose a peptidic sequence that we wanted to have four objectives. So for the first one is that it can auto-assemble like a micelle. The second one is that it can provide active targeting to tumor cells. The third one is a good drug delivery, and then imaging probe, so as to get a real theranostic thing. So in this sequence, you've got a part that is hydrophilic, and as well as got a RGD peptidic sequence, which is a sequence that is known to interact with integrin, which is a cell adhesion receptor of cancer cells. So with this RGD, we can think that we will have a good targeting to the tumor cells. The second part on the other side is an hydrophobic tail made of valine, and this part should help to provide from the peptide, maybe I can do it like this, from the peptide, an auto-assembly into my cells, the hydrophobic part being in the, the hydrophobic part being in the center, the hydrophilic part on the outer, and then you can maybe integrate drugs within the system. This reversibility depends on the pH. At lower pH, the peptide is in monomer structure. At higher pH, it can auto-assemble. This is theory, and you will see that we have to deal with this theory and doing other things. And then in this system, so there is a hydrophilic part, and the things that is pH sensitive is due to the lysines here. So the design of the sequence is not casual, it's on purpose. So we decided to synthesize this, and as we're crazy, as I told you, we did it by continuous flow synthesis, which was one expertise of one of my colleagues. And so we add one amino acid at a time. I don't know if you know about peptide synthesis, it's quite complicated. And it was done in a continuous flow process so as to simplify the washing step, to gain time, to use less reagents, etc. But we were crazy because, as you will see, it's very complicated. So first, we wanted to develop a way of characterizing these systems with commercial peptides. So we bought this sequence of peptides from an academic platform. And so we used electrophoresis to verify this system. So we were in the monomeric form. And so what do we have by capillary electrophoresis with a simple UV detector, which is not expensive? We see first what we call a neutral marker, which is important for us to determine mobility. I will not enter into the details. And we see this peak. That could be positively charged species, maybe peptides. But with UV, we don't identify the molecules. So we went further to coupling with mass spectrometry, where we get more information what is under this peak. And with mass spectrometry, we could evidence that this peak here is the one of the peptide. Okay, commercial peptide, we are sure that we have got this peptide. But by analyzing further this mass spec, we can go to extracted mass spectra, and then we can identify that there is a complete sequence that we bought. So the people did it correctly. But if we go more deeper in the decomposition of the total ionic current, which is a process in mass spectrometry, we could identify that it was only 96% pure. It is what the provider told you. But we were able as well to identify the missing sequences in the 4% that were missing. Okay, so we've got the good information on the system. So we can determine the purity, we can determine the impurity by using capillary electrophoresis, that means a separation, an efficient separation, and mass spectrometry, which is a very efficient detector. Okay, now we went to our own synthesis. So it's many steps. I will not enter in detail with coupling cycles and then deprotection, so to have this sequence of peptides. So it's quite complicated. In batch, it's quite quick. So what we did is a first analysis of our first test. And we did NMR spectrometry with 2D COSY. We did MS-MS spectrometry, so only this characterization. And from this, we could evidence that it's too complicated to analyze all what is under these peaks. So we identified by NMR and MS only 3 truncated sequences. So we can identify the sequence that are missing. Okay, now, if I go to more complex system, I need a separative method. So in this case, what we did have, you've got the commercial peptide by CEUV, the batch synthesis, the continuous flow synthesis, so we are dealing with this. And when we did go to mass spec, so in UV we see like 2 peaks, in mass spec we see much more peaks. And if we can enter in the details, we can identify all the peaks, so all the peaks with their purity. And with this methodology, we can separate the different peptide sequences, we can determine the purity and identify what are the missing amino acids. And it has a higher potential for identification. We could identify 6 sequences versus only 3 sequences by NMR and MS. So the potential of separation is very important to have a very deep understanding of what is my mixture. Okay? Then, I will not go in all the detail, we optimized the synthesis condition, we changed the residence time, we doubled the quantity, we did an overcoupling agent, etc. And at each step, modification of the parameter, we could gain in purity of the expected peptide sequence with 82%. And 82% is quite good when you look with commercial providers of peptide sequences. It's a very good result, and we are able to indicate which are the impurities. So we were very happy with this, and this shows that we gain from our expertise. To remind, my group is synthesis, electrochemistry, analytical sciences, and imaging, and here we show a very nice synergistic cooperation between organic chemistry and analytical processes. Then, as I told you, we want this peptide to be a probe as well. And so we have to provide a probe on the surface of this micelle that will be created. On the peptides, we've got amino and acid moieties. It's difficult to have specific functionalization on one of these functions. So what we did, we changed the sequence of the peptide and we add a cysteine. This cysteine has got a thiol function, and thiol function allows us to have a specific functionalization. From this thiol, we could functionalize with a complexing agent that is named DOTAGA. Maybe some of you that already did MRI analysis, you heard about DOTAGA. And then this DOTAGA, this function here, encapsulates the gadolinium, which is the MRI probe. And so we did the functionalization, and in the same time, we measured by CUV all these systems. We could evidence that going from a positively charged, negatively charged, and neutral charge, we've got the product with remaining initial reactants, that is the DOTAGA and the P2. Then when we went to mass spec, we could identify again that there were remaining reactants. So the peptide functionalization was efficient, but there were remaining reactants. Now that we had these systems, we wanted to go to micellization. That is, the peptide that we did synthesize, functionalize, should be like a micelle. But our problem is, If I look at the micellization, and here it is the gadolinium. Gadolinium is quite big. So if each peptide has got a gadolinium, it will be very difficult to make a micelle. So what we decided is that we make a mixture between the peptide that is non-functionalized and the peptide that is non-functionalized. And we want to do it very easily. So what we did is during the synthesis, we do the same synthesis, but only with a small percentage of these reactants, so that we had more or less 10, 15, or 20% of functionalized peptides and 80, 85, or 90% of the non-functionalized peptides, so as to have got some gadoliniums on the nanoparticle, but without perturbing the micellization of the system. And so we did follow this by electrophoresis, so we could quantify the quantity of functionalized peptides according to the quantity of non-functionalized peptides. And so we could control totally the thing, even by UV. And so we could do it in one pot. So in one pot, we've got the non-functionalized and functionalized, and then we can go to the formulation of the nanoparticle. So here, gadolinium complexation, okay, it's the following. So for the gadolinium complexation as well, we could verify by CEMS each step of the synthesis. So we are sure of the purity of our sample. So we've got a mixture of non-functionalized and functionalized peptides, and now we want to go to formulation. So what is a cell alpha assembly? As I told you, there is a hydrophobic tail, a hydrophobic part, and it should aggregate like a micelle. So what we did is first to do DLS. What is the size of the aggregated nanoparticle? They've got like a diameter of around 50 nanometer, which is correct when we want to go to theranostic. The second is by TEM or cryo-TEM, because we are with peptides. So we could identify it's not micelles, it's not spheres, it's nanorods. And this was new, was not shown in the literature. So we could, through this analysis, determine the length and the diameter. And if you see, DLS measures a medium size. And in fact, it measures the length rather than the diameter. So there is already a complementarity between these methods. Then we did circular decoys so as to see how the peptide interact together. I will not enter in the detail. And then, okay, we did prove that from the peptides, we could deal with nanorods. These nanorods, we could determine the length. They are like cylindrical micelles, and they are autoassembly by beta sheets. It's a way of peptides to assemble. Okay, we could determine the physical state of these nanoparticles. But how does it work? Then we went to electrophoresis. I remind you, maybe I didn't say it very well, but the monomer of the peptide is stable at an acidic pH. The nanoparticle is formed at a basic pH. So what we did is separating by electrophoresis in a basic medium, this system in basic pH. So we've got the red peak. Now, if I inject the monomer of peptide in this medium, I've got the blue peak. What does it mean? It means that there is an auto-assembly within the capillary. That means there is a fast auto-assembly kinetics from the monomer to the micelle. I inject the monomer in the capillary, and within the capillary, it makes a micelle. So formulation is made within the separation. And it's made very quickly because we've got the same mobility. Maybe it can give us the idea that it is a new way for an automated formulation of these nanoparticles, doing it in a capillary under these conditions. And then we did something that is named Taylor dispersion analysis. I will not enter in the details. It's a specific methodology by capillary that allows us to determine the dimensions. And when we could verify that we've got the same dimensions as in the previous DLS and TEM methods, and we could see at pH 5 or 3, It's monomer, so it's the size of a peptide, and at page 11 is the size of a nanostructuration. And so this allows us to show that we have complementarity between all the methodologies. Then we went to the other things with the gadolinium functional as nanoparticle. We still had nanorods, more or same in the same size, and we had the same formulation within the separation. And so we could have co-assembly nanorods even if we have the gadolinium, which is very important because as gadolinium is a complex, is a very big structure. It does not impede the formulation in nanostructures, and this formulation is already very fast in the same way. And then we went to MRI. I'm not a specialist of MRI, but what I can say to you is that we did have good MRI constraints with dependence in size, okay? So, to finish, here, okay, only to show you what we did in this example is to use capillary electrophoresis with a simple UV detector of a mass spec at each step of the synthesis and complemented with DLS, TEM, secular dichroism, and MRI to identify the structure and to provide nanorods that can be theranostic agents. And so, to finish, I will not present this. This was a proof about drug loading and delivery. But to finish, sorry, I went too quick. Okay, what I did show you I have to go very slowly. So what I did show you is through two examples, I didn't have time to show the third example, is that electrophoresis can be very interesting for developing tools for biomedical and as well environmental applications, what we did, for example, with nanoplastics that we are studying. The thing is, it is very useful because it has a very good separation efficiency. In a nanoparticle medium, we've got a polydispersity that we can discriminate within this separation. We can perform physico-chemical characterization, interaction characterization. But so as to gain performance in electrophoresis, we have to have efficient detections, UV, MS, fluo, but we have to go deeper. So now what we are doing is we go in microchip formats, and we will develop, so we are going to develop coupling with multi-detections. In one microfluidic, we'll have UV, mass, DLS, CERS, etc., if it succeeds, so as to gain again in the physico-chemical characterization of the nanoparticles. To finish, I would like to thank my colleagues. So Fanny Dorlier, Bitwi Dohan, which is a specialist in MRI, Laura Trapella in nanoscience, Gonzalo Ramirez for the PhD, Fernando Espinola, I didn't present his work, and Alisam did all the work on the peptides. And I would like to thank my colleagues in Paris, Nathalie Minier and Cyril Richard, with which we work with Gonzalo, and Minerva and Silvia from Universidad de Guanajuato for the work with Fernando that I could not present. And I thank you for your attention. And I am ready for any questions.
Okay, guys, so we have some minutes for questions. Any questions?
So thank you, Anne, for your presentation. So I have a question about the generalities of, have you been able to establish a general connections between your mobility in this microfluidic device and what have you been expecting in vivo experiments? I mean, you showed results with 2 proteins, but can you extend that observation for a more general scenario?
I don't know if I understood correctly your question, but we develop microchips for proteomics and as well for genomics. It means in one microfluidic system, we are working on this. It's a very interesting work. We are working so as to make cell lysis, protein digestion, and then separation of DNA from protein, all within a microfluidic system. And then for the proteins that are digested in peptides to go to MS.
Yeah, I'm sorry, I said microfluidic, but it's in the microfluidic. And the one you use for storing the charge of with the electrophoresis,
yeah.
the one that you showed, you did an experiment. How is the mobility of your nanoparticles when it's binding these two proteins? And I was wondering if you have a general scenario in which you can relate the experiments in a channel, in a very small channel, with what you've been expecting in vivo.
Yes, I mean, as I told you, the channel, either in capillary or in microfluidic, is the same dimension. So what we see in classical capillary, we can see it in microchip. Now, the flow rate, according to the electric field, is compatible with the flow rates of the blood. So it means we can mimic the dynamics of interactions within a blood cell. That's why we introduce the proteins within the whole capillary, and then we inject the nanoparticles so as to mimic what occurs when injecting the nanoparticle in the body, with the same flow rate, with the same vessel dimensions. As I said, it's very complicated to analyze more than one protein, but we've got a project with Gonzalo and Andres to go further than one or two or three, maybe three proteins.
Yeah, thank you.
Yeah.
Thank you very much. It's a very interesting method for quality control. And I'd like to know, in your experience, what is the minimal uniformity you need before going to in vivo or even in vitro studies?
It's a very interesting question because there is no law or decision. What we try is when we see that there is a potential for targeting that we can do with cells culture, for toxicity, for biodistribution, mimicked by electrophoresis, then we go to animal testing. So there is no generality. It means if the properties seems very interesting, we should go. I don't know how it is in Mexico, but in France, it's more and more difficult to get the liability to do this animal testing. So it's a matter of ethics, and I totally understand. That's why we try to share. But for now, we go mainly for this physical-chemical characterization and cellular toxicity test and so on. And it's still difficult for the companies to go to the maturation of these systems. There are some objects, but not all.
Thank you, Anne, for your presentation. And I have one question, and the second one is like, I want your opinion about something. The first question is related to you show us a very interesting technique to distinguish the interactions between peptides, aptamers, and so on with nanoparticles. But what about the behavior, the changing behavior of the nanoparticles once it's inside in a fluid? For example, in media, you have human serum albumin conjugated with your nanoparticle, and then you put it in the cells to interact. And then there is a changing. How can we control or try to mimic what is happening on cell culture and then try to mimic that on the in vivo?
Okay. We've got a project that is more or less in this domain to couple this microfluidic analytical methodology to an organochip or cell on a chip. I don't know if you heard about this. So, and then we can separate according to the nature of the compounds and then see what it implies on cell internalization and so on. It's a project that we have, it will take some time, but we are confident with this to complement this pure analytical system with a more biological system for gaining all this information. And as your colleague said, for now we don't have this, so we try some nanoparticles by MRI.
That sounds interesting. The second one is, I would like to know your opinion about. I all the time believe that protein corona, let me rephrase this, that nanoparticles are able to bind not only to protein, but also to lipids and carbohydrates. But because of the techniques that we are using to study them, we just purify proteins. So in your experience, maybe it's not the scope of your investigation, but what do you think about this thought? Do you believe that there are some other, besides proteins, but also lipids and carbohydrates bound to these nanoparticles? Yeah. And what is the implication of this?
Yeah. What we do with proteins, we can do it with any other type of molecule that you want to study the interaction. The only thing, as you saw in some examples, is there should be a mobility shift, or there should be a depletion, but is dependent on the concentration, so as to have more information. So it can be any type of S plus L interaction. Another project that we have is about nanoplastics. Nanoplastic is a very emerging, not emerging because it exists since many times, but known now and identified now, very emerging pollution. And nanoplastics can enter in the body and can perturb all the things, proteins, modification, and pathologies, etc. And with this, we use we study interactions of these nanoplastics with proteins, but as well with pollutants, organic pollutants, PFAS, and others. And so we can develop it for any type of compound. I mean, and for nanoplastic, it's not called protein corona, it's called eco-corona. And I think eco-corona is more representing what we want to do than only protein corona, which is restrictive. You're welcome.
Thank you. Thank you very much, Hans, for your great talk. I think apolipoprotein E is an Alzheimer biomarker, right? So I was wondering if you are also working on Alzheimer diagnostics.
Yes, I didn't show it, but you mean biomarkers. Okay. Protein can be a biomarker in some extent, but we deal with other biomarkers. What we deal with diagnosis for biomarkers is to develop a microfluidic system that allows for biomarker pre-concentration, because they are in very, very low quantity. And so at the beginning of the microchip, we develop a strategy for sample pre-concentration, then separation for electrolysis, and then detection. But we can implement as well with increasing the limit of detection of the samples. And at each step, I didn't have time to show you, we use aptamers. Because aptamers can capture the biomarker very specifically. Then we can release it by increasing the temperature. And we know how to increase the temperature in the macrosystem. Then the separation, if there are many biomarkers, maybe 3-4 biomarkers identifying a certain cancer, for example, we separate them, and for the detection, we can use electrochemical detection with aptamers that increase the sensitivity. So that's why my talk spoke only about nanoparticles, but we work a lot with ligands and with aptamers at each step for sample pre-treatment, sometimes sample separation, and then detection. And so we work on all these aspects. We work on very technological development, but always going to the fundamental of what is behind the results. So it's our way of working.
Questions?
Thank you, Anne. Thank you, Anne. Quick question. So 2 questions, actually. It's related more in general. If you could tell us what is the end product that you aim of producing like through all those development, well, as you say, you have all those technological development, and if you could give us some example of products you aim to, but you would like to commercialize eventually. And secondly, in general, lab on a chip, or this technology has been around, or this term has been around for a while now. It has been hard, from what I heard, from what I know, to get to the commercialization stage. What do you see as the challenges? Why has it been so hard, and do you think it's going into the right direction now?
Okay, remind me the first question. The first question was the, okay.
So what is your end products that you would like that you are aiming for? Like,
we are mainly in a fundamental point of view for the studies. So we are very at the front of the investigation. We know what is the application needed. So we know which properties we need. For now, as I told, the companies dealing with diagnostic don't go so much with nanoparticles. It's a challenge for the academic members to prove that it can be interesting. But maybe it's not the most interesting because of toxicity of the nanoparticles. And when you inject nanoparticles at one moment, they will go away from the body. What is their impact on the body? And for example, we work with gadolinium, which is the most known contrast agent for MRI. But now, if some of you, and for example, I do many MRIs in my life as a patient, and now in hospital, they try not to use gadolinium. Why? Because when gadolinium is injected, it's complex, so non-toxic. When you release it, it can decomplex within the environment, and it's very toxic, gadolinium. And so now they try to improve the apparatus for MRI, the quality of the images, so as not to inject gadolinium. In our lab, what we do is try to change gadolinium to another metal. And so we are working with iron, for example. And we are trying to change the paradigm from gadolinium to iron with another types of nanoparticles, which are graphene carbon dots, I would say. We don't know exactly the structure. And so we want to integrate iron instead of gadolinium. So, as I say, we are at the front of investigation, trying to show that there are other possibilities. Changes gadolinium to iron, okay, etc. Or going to peptide nanostructure. It's not so common. So, and then the companies want or do not want to go, but we are to show where the innovation can be. It's more on purpose. Now going to lab on a chip. It's not new. As I say to my students, lab on a chip is not always necessary. It depends on the application you want. Lab on a chip can be transportable. So if it's in the field, you want to make a quick analysis, okay, wonderful to use lab on a chip. Or else, maybe a simple classical chromatography or electrolysis is sufficient. So it depends on the application that you want to do. This is one thing. The second thing is, I'm dealing with plastics contamination, but my chips are made of plastic. So I'm a little bit crazy. So what we do is, with colleagues in Brazil, we try to know how to recycle these plastics that are making the lab on a chip. The second thing is we are changing plastic to paper. So we are doing lab on a chips on paper. So we are always thinking of something new, always taking in mind toxicity, pollution, circular economy, if we want to see it in this sense. So lab on a chip can be very, for example, what I said about proteomic and genomic, this can be, from an applicative point of view, very interesting, because you can have access to the DNA and proteins at one time. And this can be very interesting. So lab on a chip is not always useful. It can be very complicated, and as I told you, we have to find new detectors and combine them, so it's not so I wanted to tell you everything about this, but I don't remind. Sorry. And for example, when we deal with nanoplastics, what we want in microfluidics is to have a high throughput. And this is very important because nanoplastic, it can be many types of polymers, sizes, functionalization, porosity, etc. And so we need a high throughput analysis. So I hope I did an answer to your question.
Okay, is there any other question?
Thank you for your presentation. You talk about the diagnosis, application, and biodistribution of these nanoparticles, but what happened with the elimination? Are there bioaccumulation of these nanoparticles? Are there another toxic effects during the elimination?
Yes, that's why it's a very interesting question. I didn't speak about this. There are many literature, it's not my specialty, but there are many literatures that try to see how is it integrated and destroyed within the cells. It can be released as such, as a nanoparticle, or it can be disaggregated, then dissolved within the cells. So it's something that is very important to understand as well, but it's more a matter of biologists. For peptides, for example, why did we go to peptides? Because we hope that when they are released, it's peptides. So it should not impact so much. But in fact, I'm not sure. When it is in the environment, it can be complex to any type of compounds. So we have to see the whole line of the life of these objects. And so we deal as well with graphene dots, so carbon dots. And so we think, okay, carbon, so we have it in the environment, so maybe it's not so impacting until there are tests to verify that it does not impact. But the transformation within the body and the effect when rejected has to be as well a matter, but it's not my specialty.
Thank you.
You're welcome.
You have other questions?
Okay, so before thanking Anne again, Anne is going to spend all the week here. She's going to give a 3-day course, so please approach her, ask her a lot of questions, make the most out of it. This, as Gonzalo said, is not only a very good scientist, also a very good human being. So let's thank Anne again for her talk.
Muchas gracias. I did my talk in English because it was more easy for me, but I speak Spanish as well. So for the students, you can come and discuss with me in Spanish. It's not a problem.